All right, so we're going to talk about enzymes today. And before we do, I would like all of you to draw me an example of an exergonic reaction. So if you can make your X and Y axes, I would like you to do an exergonic reaction for me first. I'll give you about 1 minute to label it. I want you to tell me the reactants, the products, the change in enthalpy, and the activation. All right, there you go. That's what you should have. Now, I've already given away that this is an exergonic reaction, but we're going to expand on that. Exergonic simply means that energy is exiting the system, or the reaction. Now, take a look at the enthalpy. Is this reaction gaining energy or losing energy, Mark? It's losing. So would you expect this to have, would the energy content be at a loss or at a gain? And how do you represent a loss? As a positive or as a negative? Correct. So the change in enthalpy, oops. The change in enthalpy is a negative value. And here's a little tip. It's a little hint. It's a little cheat. Whatever the enthalpy change is, the Gibbs free energy will be the same. So if delta H is negative, delta G is negative. And Mark, if delta G is negative, does that mean that this reaction is spontaneous or non-spontaneous? That's right, spontaneous. In a spontaneous reaction, there is no energy added to the reaction. It's going to happen on its own. You get your hair cut, your hair falls to the floor. You're sitting on a paddleboard, you're going to go with the current because you're just going to sit there. Now finally, which of the two parts of the reaction would you consider to be high energy when compared to the other? Are the reactants high energy or are the products high energy? Mitchell? The reactants are high energy, and how would you consider the products? So you're going from high energy to low energy. Is that catabolism or is that anabolism? Think about it. That is catabolism. I like how you stopped and thought about it. You don't have to immediately go, catabolism. It's not a race. I want you to stop and think about it. So this is catabolism. Catabolism is the breakdown of high energy molecules into low energy molecules. That pretty much spells it out, guys, when it comes to a reaction like this. And I was a silly Billy and I didn't label my axes. Be sure you always do that. On the AP exam, that actually can be worth points. Just label your axes. Now, what I would like you guys to do next is I want you to create an endergonic graph for me. Draw me a graph that represents an endergonic reaction. I want you to label the reactants, the products, the change in enthalpy, and the activation energy, please. I'll give you 1 minute. Stuck. All right, here we go. So we have our reactants, R, products, P. Wait, I did that wrong. What a doofus I am. Got a little carried away, didn't I? There we go. That's better. Okay, let's try that again, shall we? Okay, the difference between your reactants and your products is going to be your enthalpy change. And the difference between that energy spike, the activation energy, and your reactants is your energy of activation, EA. This very top, guys, is called your activated complex. So the very, very tippy top of that energy spike is called your activated complex. The energy difference between your reactants and your activated complex is called your energy of activation or just activation energy. Okay, let's pick on somebody else here. Eeny, meeny, miny. Tiozo, is this reaction gaining energy or losing energy? Correctamundo. So would you expect the change in enthalpy to be positive or negative? That's right. And if the change in enthalpy is positive, the change in Gibbs free energy is positive. So is this reaction spontaneous or non-spontaneous? You've got it. You're on fire. So the first thing I want to make sure we get written down is this reaction is endergonic. This means energy is entering the reaction. Next, if delta H is positive, then delta G is positive, and that means the reaction is non-spontaneous. This term means, or this, this would mean that this reaction does require or experience energy input to the system. All right, finally, as she said, this is the products have more energy than the reactants, so this is going to be anabolism. This is the building up. All right. So that was really pretty much a review of everything from yesterday. Now, let's get into the new stuff. Are there any questions based on yesterday's class? Chris, you good? All right. So today we're going to talk about enzymes. This is a continuation from yesterday on YouTube. This will be called lesson number 13, part 2, Thermodynamics and Enzymes. So enzymes, what the heck are they? Enzymes are biological catalysts made of protein. If it's biological, it is in living things. Enzymes are found in animals, plants, fungi, protists, archaea, and bacteria. All living things. Catalysts basically speed up a reaction. And when you guys see me write RXN, that's the abbreviation for reaction, they speed up a reaction by lowering the activation energy, or the EA. And then a protein. Well, a protein is a macromolecule. Where are proteins made? When I say blank, you say protein. Ribosomes. What are proteins composed of? What are their monomers? They're composed of amino acids. Okay. All right. Now, another little factoid about enzymes. Most enzymes are named after what they metabolize. And they end in what suffix do most enzymes end with? A-S-E. They mostly end in ACE. So the things that they metabolize are called substrates. And what I want to do is I just want to share with you guys a few examples of enzymes and substrates and how they get their names. So the first one would be amylose. This is something you got from Unit 1. What is amylose? A carb, lipid, protein, or nucleic acid? It's a carb, okay? Is it a monosaccharide, polysaccharide, or disaccharide? Unit 1 question. I told you, the further away we get from Unit 1, the harder it's going to be. It's a polysaccharide. Its nickname is starch. The enzyme that breaks down amylose, and amylose is found in crackers, cereal, bread, grain, noodles. It's amylase. See how that works? For those of you that have taken psychology or are currently in psychology, you may have heard of this word, acetylcholine. It's what allows neurotransmitters to enter into a synapse during the synaptic gap. The enzyme that gets rid of when there's excessive acetylcholine, so you're not overstimulated, is acetylcholine esterase. Big word. Another one, lipids. They're metabolized by lipase. Yeah. A substrate is the molecule or the compound that is metabolized by the enzyme. Then lastly, peroxidase. Or excuse me, peroxide is broken down by peroxidase. Okay, so that's just a few examples of some enzymes. There's plenty more. Transcription is done by transcriptidase. That's just another example. Okay. Now, the next thing I would like to go over with you all is an enzyme substrate complex. What happens when the enzyme and the substrate actually come together? Okay, well, I'm going to do a lot of drawing here. So here we go. Let's say that is my enzyme. It is that specific shape. The area where a substrate connects with an enzyme, like Legos, is called the active site. This is called the active site. That is where an enzyme and a substrate come together, like a key and a lock, your foot in your shoe, this marker from my computer into its little port on my computer. That would be the active site. And then you actually have your enzyme. Or I can flip-flop, your substrate. This is your substrate. This is the substance that is actually going to get, in this case, broken down in a reaction. So when they come together, you actually have an enzyme substrate complex. This is where the enzyme is going to catalyze the substrate. This substrate would have broken down on its own, but this is going to speed it up by quite a bit. And so by the time you're all done, what you have now is your enzyme and you have your products. This is known as an enzyme substrate complex or enzyme substrate interaction. Does it appear as if you're converting a large molecule, a high energy molecule into a low energy molecule, or does it appear as if you're converting low energy molecule to high energy molecule? This is high to low. So what type of metabolism is this? This is catabolic. Well, what if I went in the reverse direction like this? I went from here to here. What type of metabolism would that be if you went from right to left? That would be anabolic. Guys, this works both ways. You know, Chris looks like a pretty strong guy. Let's say he goes to the gym today. He's really going to tear his muscles up really good. He's going to work out really hard. The way that people gain muscle mass is twofold. They work out a lot, but then they feed their engine, fuel the jet. They got to eat a lot. If you saw the amount of food that Dwayne Johnson eats in one meal, it's probably more calories in one meal than what you guys have in 3 days. Because his muscles demand that type of caloric intake. And so Chris is going to work out really hard after school today, and he's going to just shred his muscles. And that's what happens when you work out. You actually shred and damage your muscles. And when they heal, you're hopefully going to have them back bigger and stronger than what they were before. But you have to eat a lot of food. You got to have a lot of calories to get that done. And so he's going to go to Burns Steakhouse and I get a couple big, fat, juicy, expensive steaks. What is the main macromolecule that you would expect to find in steak? Carb, lipid, protein, or nucleic acid? Protein, right? Lots of protein. So he's going to eat these proteins, these steaks. And his digestive system is going to break them down into their amino acids. Is that right there catabolism or anabolism? That is catabolism, breaking it all down. Well, those amino acids are then going to diffuse into his bloodstream. And there's a lot of amino acids because you just ate 2 huge steaks. And they're going to go to his muscles. And they're going to help repair his muscles and make them bigger and stronger than what they were before, building them up. Is that catabolism or anabolism? See, it happens both ways. In us, it goes both ways. This is also known as secondary production. You've likely heard of plants are primary producers. They produce the main energy for an entire ecosystem. But when you eat the food, you guys are heterotrophs. When you eat the food and your bodies get bigger, that is called secondary production. You're making your own bodies bigger. And so catabolism and anabolism, it's not just one or the other. Both forms of metabolism happen in all forms of life. He's going to digest it into its small pieces, and then it's going to get reconstituted into him, his body, into his muscles and all that to build him up more. That's anabolism, okay? So in order for this to happen, we're going to talk about the next topic. That is going to be called collision theory. So in collision theory, here's what happens. Let me tell you something. Something I've been seeing a lot in class. Watch this. Chris, I've been seeing a lot of people do this. Hey, man, yo, what's up? Don't touch me. There's a pandemic going around. I see so many kids in school doing this. Hey, yo, what's up? Really? Don't touch your faces after you just and then adjust your mask. Great. So what I'm getting at here is, you see a lot of this. I want to make this sound good. That. You know, you guys probably like slap hands before you miss, you go, oops, or you do this. That's pathetic. You want to make that big clap sound. So that's the product I want. That is my product. I want this. Well, let's do it again. That's what I want. There's 2 things in order to make that sound. One, my hands have to go fast enough. I can't do this. Can't do this. Can't do this. That hurts. It's too much. That's good. Second thing, my hands have to be oriented in the proper position. I can't do this. That's not the right sound. How about this? No, it's not the right sound. Maybe this. No. How about this? No, that's not what I'm looking for. There it is. So when you want to have an enzyme and a substrate react with each other to form a product, 2 points of criteria. 1, the enzyme and substrate must be moving at an appropriate velocity to stimulate a reaction. If Mark gets in a fight where he wants to hurt a guy, he can't do this. That's not going to hurt somebody. You got to harden it, velocity. Second point The enzyme and substrate must be oriented in their appropriate configurations to stimulate a reaction. So when I want to make this sound, my hands have to be positioned like this. Not like this, not like this, not like that, not like this, not here, that. Same thing goes if my fist is a substrate, my open hand is an enzyme. They have to be oriented in the proper position, and they have to go fast enough. That's the two things you need when you have collision theory and in order to make an actual product or products. All right? Now, okay. What I would like to do with you now, I want to do a simulation. We would have done this if we weren't in the middle of a pandemic and we had to wear masks. But if this was a normal year, I would have a box of Ritz crackers and I would pass the sleeve of crackers around and I would ask all of you to grab one cracker. So let's just simulate this, okay? So here's your Ritz cracker. What type of carbohydrate, polysaccharide that we discussed in class, is this Ritz cracker made out of? What do you think? Amylose, very good. So this is composed of amylose. And I already said this is a polysaccharide. So this is made up of a huge sugar chain. And what I'm going to ask you to do with this cracker is I would ask you to place the cracker in your mouth, but don't chew it. Don't crush it with your tongue and your palate. Just let it sit there. And eventually you guys are going to have some really grossed out looks on your faces because you're not used to that. You're used to eating a cracker immediately, not just letting it sit around in your spit. So it's going to sit there for about 2 or 3 minutes. And some of you are going to like, all right, can I eat this thing? This is disgusting. I say, no, you got to let it sit there for a minute. What's happening is that your saliva is starting to metabolize it, to break it down. Because within your saliva, you have an enzyme called salivary amylase. Carbohydrate digestion begins in your mouth. And it is going to get broken down into maltose. Just by looking at maltose, or if you recall from Unit 1, what size of a sugar is this? Very good. It is a disaccharide. So here's a question. Is a disaccharide sugar small enough to diffuse through the walls of your intestines and go into your bloodstream? No. Can you go smaller than a disaccharide? You bet. So after you swallow this big goober of spit and amylose, or actually now it's maltose, that's going to go into your stomach. And it's going to pass through your stomach into your intestines. And in your intestines is another enzyme that is going to break it down from maltose to glucose. Glucose is a monosaccharide. This enzyme is called pancreatic amylase. Your pancreas does not store food. What your pancreas does is it produces a lot of hormones and a lot of digestive enzymes to help metabolize food faster. Now, would this cracker have broken down in your mouth if you did not have salivary amylase? Yes, it would. But I mean, would it happen fast or slow? It would be so slow. Would it be fast enough to provide you with the energy needs that your body requires? Nope. That's why we have enzymes, because enzymes can help metabolize food and reactions that give us energy quickly. We're mammals, we're endotherms. We need energy fast. If you have a pet snake or a pet turtle, they're reptiles. They can eat once and be fine for days. Mark is barely fine after 2 hours. He's a mammal. He needs food. He needs to keep that internal body temperature going. And that's fueled by the food that we eat. Enzymes are able to take the food that we eat and convert it to energy for our metabolism. Yeah. People that are lactose intolerant don't have lactase. The bacteria in their guts don't have lactase. And in fact, if you didn't know this, by far the minority of the world does drink milk. The majority by far is lactose intolerant. Because think about it, how many cultures in the human history actually drink milk? Not a lot. So if you are not lactose intolerant, you are the definite minority. The majority of the world didn't do that. So there's that. It's a mutation. And yes, that is a good point. Lactase breaks down lactose. All right. So moving on. What I'd like you guys to do now is, once again, I want you to draw for me an exergonic reaction. But we're going to do something a little different with it this time than what we did earlier. You don't need to label it. Just draw it. Okay. But here's the only label I want you to call it. I want you to say this is without a catalyst. This graph is an exergonic graph without a catalyst, or an exergonic reaction. What we are going to draw now is an endergonic reaction, but here's the catch. Let's say that this dotted line I'm drawing is your body temperature. Your body temp is about 98 degrees Fahrenheit. In order to make this make the product of this reaction, you've got to get it up to 105 degrees Fahrenheit. If your body got up to 105, that's going to be borderline hospitalization. That's not That's not safe. But you got a problem. You need to make the products of this reaction, and the reaction requires a temperature of 105 degrees Fahrenheit. So are you going to be able to make products? Maybe a little, but nowhere near enough what you're supposed to. So what you're going to do, enzymes to the rescue. What if I told you there's an enzyme that can be used to lower the activation energy and reduce the amount of time it takes to make the product? This green line is going to represent with a catalyst. So today I want to ask you, which reaction ended first? The exergonic reaction with a catalyst or the one without? With. Let's go to Emmy. Which reaction used more energy, with the catalyst or without? That's right. So the catalyst sped up the reaction and it used less power. It's a win-win. And we need power fast. We need energy quickly. We can't wait for things to just decompose and break down. We need them now. That cracker in your mouth will break down. It might take weeks and months, but it will break down. But with your saliva, it happens in a matter of minutes. And that's what we require. Okay, let's get to the last part for today, guys. Enzyme regulation. I'm gonna try to do the, we got about, oh, we got time. We're great. So these are called feedback loops. Hannah, do you have a job? You want to work at Hollister though, right? Okay, perfect. So let's say you go to Hollister and you are just a wonderful employee. You fold the clothes when the customers come in and throw them all over the place. You sell like at least 5 pairs of jeans an hour. Your manager comes up and says, Hannah, I really like what you're doing. You sold 5 pairs of jeans. How about 7? I want you to sell 7 pairs of jeans the next hour. And you do it. So Hannah, that's awesome. I want you to sell 10 pairs of jeans the next hour. And you do it. I want you to sell 12 pairs of jeans. You're doing so good, I want more of it. That's what this type of feedback loop is called. This is called a positive feedback loop. This is where you are accelerating what is already happening. You want more of it to happen. The Lightning won 2 Stanley Cups. Heck, I won 3, 4, 5. I want them all. The Rays won last night. Well, I want them to win tonight and tomorrow and the next night. I just want to keep winning. That is positive feedback. The way we say this in biological terms is the products of a reaction stimulate future reactions. So I say, Chris, I really like how you sit here in class, you take your notes diligently, you don't skip, you're a good kid. He's like, all right, I'm going to keep doing that. In fact, I'm going to do better. That's positive feedback. You're basically wanting to keep doing what you're doing, but you want to do it more. The classic example for this in biology is childbirth. Now, fellas, in case you didn't know, females have a uterus. And a uterus is made up of smooth muscle. There are 3 types of muscles in our body. There is skeletal muscle, the type of muscle that you work out in the gym, your quads, your biceps, your triceps, your abs, your shoulders. That's skeletal muscle, and we can voluntarily control that. We can move our body all over the place. Cardiac muscle is in your heart. You cannot control it. It is involuntary. And then the 3rd type is smooth muscle. This is what your hollow organs are made out of. Your esophagus, your stomach, your intestines, your uterus. And so when the uterus actually contracts, whether they're just menstrual cramps or they're actual childbearing contractions, they cannot be controlled. So let's say that a woman is giving birth and the baby's ready to be delivered. The first thing you're going to have are uterine contractions. Now, when that uterus contracts, look at me, the baby's head is going to push against something called the cervix. The cervix is an opening at the bottom of the uterus where the baby passes out. It connects to the vagina and then out of the body. Every time that baby's head is pushed into that cervix, something happens. But I'm going to write this down first. You got uterine contractions. Baby pushes against cervix. And for those of you that know anything about childbirth, do you know how many centimeters dilated the cervix has to be? Right now? 10 centimeters. So ladies, your cervix has to be that. So the baby pushes against the cervix. Now, here's where it gets a little intricate. A signal is sent to the pituitary gland, which is in your brain, to secrete the hormone oxytocin. Oxytocin is called the love hormone. Its levels are high during sex. Its levels are high during childbirth. It actually kind of helps the mother bond with her baby instead of saying, oh, look at my little bundle of joy. You caused mommy so much pain. That really doesn't happen. It's more bonding. If there's a hormone influx, it's not where it should be, you have postpartum. Well, nonetheless, a signal is sent to the pituitary gland to secrete the hormone oxytocin. Oxytocin travels in the bloodstream to the uterus and stimulates it to contract more. Okay, guys, so this is like a vicious circle. I'm sure you can do a cause and effect here. So, Despina, Mary, all y'all work with me here. Baby's head pushes against the cervix. Okay? The uterus is contracting and the baby's being pushed against the cervix. A signal is sent to the mother's brain to release oxytocin. Oxytocin travels in her bloodstream down to her uterus, causing the uterus to contract more. So what is the baby doing with the cervix now? Pushing more or less? Pushing more, okay? More pushing against the cervix. So more signals are sent to the brain to release more of what? Oxytocin. More oxytocin is going to flow in her bloodstream to her uterus to cause her uterus to do more what? Contractions. And that's going to cause the baby to do what? Push more, and it's just a vicious circle. It gets more and more and more. So during pregnancy, you might have a contraction that might last 5 seconds, and they're 13 minutes apart. So it's like 1, 2, 3, 4, 5, all done. But as you get closer to delivery time, the contractions are longer and they're closer together. So rather than be maybe 5 seconds long every 13 minutes, they might be 30 seconds long every 3 minutes, something like that. I'm not an OBGYN, but that's pretty much how it works. So this is positive feedback. You want to enhance what's happening. The opposite is going to be negative feedback loops. In a negative feedback loop, the tried and true definition is the product of a reaction inhibits the reaction from occurring again. Basically, whatever's happening, you want to undo it. If Hannah was jumping up and down on the table and screaming at Tanae, making him cry, I would say, Hannah, stop doing that. That would be negative feedback. I don't want you to do that. I don't like it. So if you want to put in your own words, my own words would be basically you want to undo what is happening. So the classic example of this is going to be, you could say sweating. You could say your thermostat. I set my house at 73 degrees Fahrenheit. When the house gets up to 74 degrees, the air conditioning turns on and it continues to run until it gets down to 73 again. But when it heats up to 74, the AC turns on again to cool it down. But for biology, let's do a blood sugar. So let's say that Mark drinks a Sprite. Sprite has a lot of sugar, Mark. So what do you think the Sprite is going to do to your blood glucose levels? Oh, yeah. Blood glucose elevates. This isn't very good. So let's see if those of you that may know, what hormone lowers your blood sugar? Diabetics have problems with this. Yeah. Pancreas releases insulin into the bloodstream. Okay. So the pancreas releases insulin into your bloodstream. Insulin escorts glucose into cells, and this is out of the blood, guys. So the sugar is now out of the blood and and as glycogen. Glycogen is a polysaccharide made of glucose monomers. So Mark, now that you drank your Sprite, your blood sugar elevated, your pancreas releases insulin, it stores that sugar in your cells. How would you describe your blood glucose levels now? So the sugar is in your cells now. It's out of your blood sugar decreased. That wasn't a guess, right? That was a confident, I know it. It was a guess. Well, imagine that I just had a bunch of kids come running in this class and they're going to crowd our space. How can I solve this problem? Kick them out. You put all the sugar in your body is going to then store that sugar in your cells. If the sugar is in your cells, is it in your blood anymore? No. So effectively, what did you do with your blood sugar levels? You lowered it. So now, blood glucose levels are lowered. When your body temps get too hot, what does your body do? Sweat. That's negative feedback. When you're watching television in the living room too loud and your mom yells at you, what does she ask you to do? Turn it down. That's negative feedback. if you were watching television and your mom said, hey, turn that up, what would that be? Right. Positive feedback means I like what you're doing, do it more. Negative feedback is I don't like what you're doing, stop. You're going to undo it.